This is just to give an impression that with this screening approach we now started to use also on other genes, on disease-related genes. In a collaboration with Jürgen Knoblich's lab, we, for example, used this pooled single-cell CRISPR perturbation screen on 36 autism risk genes. And again, we could assess how cell states are maybe more or less present based on these perturbations, and we could actually find an ASD-enriched gene regulatory network that was, all these perturbations were enriched in all these genes that are linked to autism. There seem to be common mechanisms. And then at the same time, we are also working on periventricular heterotopia associated genes to knock them out and find common mechanism and distinct mechanisms. So I showed you how we can use single-cell technologies, especially genomic technologies, to learn more about human brain development. But of course, the big question is in the room, how similar is actually this human brain development, brain organoid development, to the real development of the human brain? Here you can see now cross-sections, just DAPI of organoids. I mean, yeah, they look quite different, and yeah, so how can we assess the similarities? Well, there are reference atlases out there for the primary human brain. We have, for example, from Stan Linnerson's lab, a great single-cell transcriptomic reference of brain development from gestational week 5 to, I think, 14. So this is available, but what about the organoids? If we look at organoids, there is actually tons of papers out there that publish single-cell transcriptomic data of organoid development. And these use unguided protocols, guided protocols. What to do with that? Ideally, we have actually an integrated atlas. We ideally would want to put all this together. And how can we actually do that? So this is the question that leads over to Fabian's part.
Thanks. So this is our transition. Thanks, Barbara. So you saw in this beautiful presentation this vast amount of data and the complexity of the particular questions, and in particular this mapping. It's really something that we can maybe sort of use math and physics, or if you want to be fancy about it, use AI for that. And you see, this is what we spent like the whole afternoon now preparing. So we use AI now to help us integrate these data and potentially superimpose them. And I want to just briefly explain just a little bit what these concepts are before then Barbara would then continue and then show what we actually find in that integrated organoid atlas data set. So just taking a step back, what is AI, machine learning? I'm sure many of you have heard about it. I mean, I guess pretty much everyone has been now playing around with ChatGPT and so on. It's maybe not so exactly clear what's going on there. But classically is, if these dots would be data sets, would be samples, right? The very classic thing is just separate, just tell me what's different between them. Give me a classifier. So this is called supervised learning. So just want to be a bit educational here. And this is discriminative learning. And once you have such a separating line, this could be as simple as in this 2 dimensions, just this hyperplane. So this is really something you can do sort of in high school, I guess. Then if you have a new sample coming in, this is blue because it's on that side of the line. So this is how a classifier works. This solves hard problems such as cats versus dogs, images, which, I'm not sure if the organoids can do it, but very early kids, babies, don't know how to do this thing. But teaching this to a computer has been a very hard problem. Has been raised in the 60s, and computer vision took 50 years to actually be able to address this hard challenge. And why is it possible now? Well, because of method advancement, these massive sort of high-performing deep learning methods, but then in addition also this massive amount of data. So particularly, for example, this classifier here, this was just possible because a huge database such as ImageNet was called, has been put together. And as a third ingredient was this high-performance compute access that we only just have now. So this is what's happening now, and we kind of can see how we can apply this. In our case, of course, we don't really have particular prediction. We don't have labels for a single cell or single cell and organoid. So what we typically have is called unsupervised learning. So we just see sort of a bunch of, let's say, single-cell RNA-seq samples that could be now, of course, not 2-dimensional, but 20,000 dimensional. But then we want to find some structure in there. And what could that structure be? Well, first, it could be just sort of see, which axis is actually sort of the most interesting thing happening. And if you look around here, you sort of stand in the middle and you look where's sort of most interesting might be this direction, where, those two groups sort of are differential. You could maybe also cluster them But this is sort of the thing, learn in these data sets. And what we want to do now with our single-cell RNA-seq, for example, but then, of course, later also multi-omi and maybe also spatial data sets, is to find such structure in these samples. So, for example, take in here a particular cluster of these cells, which obviously would live in much higher dimension, should be much more. You would then maybe be able to learn to name them different cell types. So this would be sort of this variation that we look for. So there's method for that around. I'll tell you in a minute how this works, but this is quite impactful and sort of interesting for us because, for example, Barbara showed this idea of this human organoid atlas. This is actually an endeavor in a wider thing, a network of more than 3,000 labs participating across the globe, or maybe members, but at least a very big initiative that's called the Human Cell Atlas, where we've been now putting together these samples across many different organs in the human body as well as model systems. And just to sort of show disease relevance this morning, right, Beate, we spoke about relevance, what's the translational impact. This is from a beautiful review from Aviv and Sarah, where they've been just aggregating what type of perturbation data sets, disease data sets are already available for the various organs. Now that we have these, you can ask a similar question. Now you have your healthy control, how would you superimpose the disease on top of that? Of course, also, in the perturbations that Barbara and her team is doing, of course, similar question would be, you have the control organoids, how can you map these on top? So the first question that I want to answer here, just briefly, is explain how we will be building this integrated atlas. So what makes an atlas an atlas? We've been looking in the lung, and this will be the example that I'll show you in the next 5 slides. And there was very similar question, just as Barbara showed. Many papers around that had a single data set, maybe often already called an atlas, but would have different variations. And typically, if you take one of these data sets, it would look different in another lab. So maybe we can take this atlas metaphor a bit further. So if we build an atlas of the world, we don't have like a single person going around and sort of mapping every street, but we have a few doing the same thing. So in this example, there was this guy going around a university campus just asking different people to draw a map of the world. So they do a job, but maybe one forgets Madagascar, the other one maybe forgets Australia. But if take all of them and sort of integrate them, wisdom of the crowds, you kind of do find Australia again. So that's nice. And it's a bit similar to what we observed. In our case, also in a single lab, the data sort of, you would find your clusters and it's kind of nice, but if you do this now multiple times, there's sort of very often the major axis of variation is lab effect, or is maybe technical, there's maybe location where you get the samples. And similar in these organoid protocols, dominant effects would be this. So you want to remove these and also integrate them. We've been coming up with metrics for doing this. We've been also coming up with a few methods for doing this. But I think the key point I'm trying to make here is now that you can use these neural networks that I was showing before, again, for this type of analysis. So what you want to do is you take a, let's say, gene times cell matrix here, and then you would sort of inform each of these neurons here with one particular transcript input, and you would squeeze them down in what here is called a bottleneck layer. And remember, we don't have particular labels, so we cannot do a prediction. So you actually blow them up again, and then you make the output as similar as possible to the input. So you compress them first, so you need to sort of throw away data. In this case, you throw away the sparseness in the data, and then you sort of fill it up in a more continuous fashion. And what's interesting is that this bottleneck, so this layer where it has been compressed, this is sort of where something interesting happens. In the simplest case, this would be like learning like a principal component analysis, but this is now a strongly nonlinear version of that. And if you now want to remove batch effect, remember, this is what we want to do. We want to sort of superimpose from different labs things on top of each other. If you now inform this neural network at the beginning that, hey, I'm from batch 1, I'm from lab 1, lab 2, well, then you can actually make them superimpose automatically, because in the bottleneck, you want to be as good as compression as possible. So you use this information. This is lab 1, is maybe always shifted by 2, so you just sort of always superimpose it. So this is how these data sets clean up. It actually works quite well. You sort of find structure. And we've been running a few competitions at the largest machine learning conference. This last year was maybe like 300, then it sort of keeps growing. More than 1,500 people participating, and I think pretty much all of the winning methods use one way or another of these neural networks because they're just so powerful in terms of scaling. So my lab has been really strongly investing to this. We've actually been, together with Nia Josef, also the first sort of to bring this into the field with the autoencoders, and this is really popular now. So let me show you what we can do now to actually learn such an integrated atlas. Again, I will show this for the lung now, and then Barbara sort of makes it beautiful for the organoids. So I know maybe not so much interest in the lung, even though they're apparently also organoids, but just stay with me. This is work by Lisa and Malte, and we did the same thing. We had 14 different data sets. We first harmonized annotation of 500,000 of these, and then also harmonized metadata at anatomical location, and then just put all of this together, and then we build this map, this atlas. In order to make it more look like an atlas, just put a circle here. And now you see your different continents, right? The epithelial cells here that you can zoom in, where you have all the detailed annotation, the endothelial and stroma cells up there, as well as the immune cells. So this is typically what these organs are made up of. And because you have this thing, you can sort of detail check what's going on where. So now that we have this thing, but we have it also represented as a projection, how can we actually use this? So how can we now map things on top? Remember, Barbara's challenge was we wanted to map the actually human development onto organoid development or vice versa. So how can you do this mapping? And I showed you just this for the lung here. This is called, in traditional machine learning, you would sort of do these things separately. And the key idea that we now just copy from machine learning is called transfer learning, where we take now one thing that we've been learned once, and then we move it to the next one. It's just as if some of you is doing whole genome sequencing, right? You would never sort of assemble by yourself again. You would just map on top of a reference. And the same thing is what we do here. So, we have, for example, here we have sort of this background distribution, this background atlas that we learned. That could be, for example, the human brain development, and then you would sort of map the organoids on top. And then you would see maybe there's some new sort of regions happening here. And how does this work? Well, in practice, we would be sort of training here on the reference, but then we would sort of keep these weights here fixed. And we would now just fine-tune sort of this frozen network. We just update very few edges, and this is how we can make things embed. All right? So here's just a very short example how we would be doing this actually now for COVID. Now we would take this atlas, take now a Curie data set for some lung lavages, and we would see that in this for COVID patients, you would see they sort of aggregate here, but then there's also some interesting aggregation here, which is split by disease severity. So now you can maybe use this atlas to actually explore what's different here, right? And because we have maps, we can actually say, oh, this region is actually macrophages. So let's zoom in what's differential between severe and moderate trajectories of COVID. And we actually see that in the moderate ones, these tissue-resident macrophages seem to be more active because of a few expressed transcripts that are so specific for this. And for example, the CXCL2 as well as the CL2 would highlight monocyte-associated macrophages, which has been also, of course, before documented to be associated to the severe situations. So this sort of just very quickly drops out of that. So let's use this also for that atlas. In this atlas, initially we started with a rather small data set, but of course, there's always more and more data coming in, and you want to sort of be more complete. And similar, this will be happening also in the organoid case. So what we now did is we took the same idea with transfer learning. We just extended the atlas. And now we were ending up with more than 2 million cells, more than 400 people, where we can do all kind of downstream analysis. And we have a few examples here. You can look for rare cell types. You can maybe map GWAS hits on top of it. And you will see a few examples what we do in the organoid case. But just one highlight here, what we found, because we have multiple diseases in there, and that I think would be interesting if you also multiple perturbations, that there's actually one cluster where a few of them co-localize. For example, in this one where SPP1 is high, which is apparently associated to monocyte-derived macrophages, this is both in COVID as well as in lung carcinoma associated, and this may be associated to some type of scar formation. Right. So last sort of technical point that we do, we've been now extending this integration type of techniques. Remember, we sort of compress and we sort of blow up again by a sort of additional factors. And I won't say now in detail what these are, but the key trick is that we don't sort of trivially embed here, but we sort of learn a more interesting embedding what each batch would be. Remember, before we just added sort of one of these nodes for each batch. Now we learn an additional dictionary of how these different data sets would be superimposing. And we can use this one also to then query for things. This is called SC-Poly. This was this integration technique that we then actually used in the organoid atlas. I spare you with the details, and maybe I also skip the actual example that we did for the lung, just to sort of be a bit in time. Well, I just go over that. And we could use this for lung and integrate it perfectly. And we also saw some sort of metastructure. This was sort of these embeddings, Barbara, that I was mentioning before, where you can then sort of see what's differential between, for example, I think in this case it was sort of alive versus post-mortem samples, and you could sort of interpret where this batch variation comes from. So basically what I'm wanting to say here is we have a technique based on neural networks that can integrate data sets, but it can also tell you where variation comes from. And this is something that we follow up now. So with this, this enables this multi-scale integration. So let's now try this out to our organoid data set. Here you go.